Good evening, BSc third year students. Now we discuss I have given discuss the induced breeding. Induced breeding is very important topic in unit second. I have given the information about the background of induced breeding in last period. What is the background of induced breeding? Induced breeding is very important articles about the and that is related to the hypophysis. All of you know the I have given information about the hypophysation and what is meant by the hypophysation. You know, in last year we also discussed about the uh, endocrine glands, and endocrine gland it is a very important gland, master gland, that is a pituitary gland. Same name, that is the hypophysis, and second also name is the hypophysation. Yes, that is the pituitary gland. What is the historical background about the pituitary gland? Uh, Pituitary gland, the number of scientists studied about the pituitary gland hypophysation adds Pickford in 1960 59 to 1964. Scientists pointed out the role of pituitary gland in the reproduction of vertebrates. Next scientist also, that is the Hossey scientist. Hossey scientist studied about the pituitary gland and find out the injection of the spawning fish. Yes, and some find out the concept of hypophysis, and that is the concept is successfully first time. Numbers are the Brazil scientist, Russian scientists also studied about the hypophysation, but the Russian scientist in 1932 uh, selected the species, that is a Strivan species, at the ripe period, mature period, and that is the some scientist also studied, Russian scientist, in mammals about the hormones. Yes, that is the uh, Giberlitsky in 1937, and find out it's related the hypophysation about the fecundity, means ripe of the eggs, how many eggs in the Strogan fishes and other species. Yes, the next scientist and other countries also, that is the USA, studied about the hypophysation in the fishes. That is the Hossey scientist, American scientist, studied and find out the injection of the hypophysation in 1940. Referred scientist and successful the all the species spine to the spawning fishes. And next scientist also, that is a Japanese scientist, Kizwiri scientist in 1948, Fujita Eatles 1948, Commento 1950, performed the successful experiment about the pituitary gland and find out its concepts and idea about the pituitary gland. Pituitary gland is very success in the spawning type of this fishes. Our countries, that is in India, also studied the spawning in induced breeding of the spawning of carps, some mammalian also, which is a famous scientist, that is a Khan scientist 1938, about the vertebrate animals, not only that is a fish but also mammals also, successfully the hypophysation. Yes, in 1955, and find out some the concept with the help of research institutes, that is Central Inland Fishery Research Institutes, about the fishery and its fishery to it's in the best example catfishes. Yes, and induced breeding for uh, pituitary gland of injections. I have given that is the name of the scientist, which is the Khan scientist in 1938 with research institutes studied and find out some concepts about the pituitary hormones in injection. In Indian scientists Chowdhury and Alkani in 1958 successful making the injections about in the major calves. Specific examples that is Lebiorohita, Sriana mrigal, yes, and other predatory fishes also. Best example Punctius, yes, it's Punctius and its genus Channa. That is some Chowdhury and Alkani scientists successfully find out the injections in the induced breeding at the time of spawning period of fishes. Yes, in Chowdhury scientists in 1958 successfully carried out the cross breeding in the 12 species of different. 12 species means some species of indigenous, some species of exotic. Indigenous species Catla, Libio, Mrigal. Exotic species grass carp, silver carp, Cyprinus carpio. And the predatory fishes, Valago to Mr. Singhala. Yes, and some other species. At these types are the 12 species collect different species and that is the male and female fertilize the eggs artificially by the artificially, yes, and that the Chowdhury scientist successfully. And Indian next scientist in 19 Ramaswamy in 1956 to 1957 successful the induced breeding. It is H. fossilis. H. fossilis means Heteropymptinus fossilis and Clarius batrachus. Accessory respiratory developed organs in this fish. This species majority found inside the mud. That is that species predatory fishes, air breathing fishes developed air breathing. That is successful the injection at the time of spawning. Ramaswamy in 1956 to 1957. Not only that is the fishes but also other vertebrates, that is a mammalian, studied the gonadotropic hormones in 1958 and tried out the different types of mammalian hormones, yes, with some species also. These types that is the supports the to include supporting the what is the important characteristics of the hypophysis to use the one happa and other that is the nets also, small nets, cast net, gill net. It is very useful for the collecting the fishes at the time of spawning, and these fishes some fishes are restless. That types of fishes collected with the help of net and give some that these types of the treatments or supply food and to separate the ponds. That is sometime that is the fishes hatching period. At the time of hatching, that is happa use, net also use, and to separate the fish. If the temperature rises suddenly, if the physical characteristics and chemical characteristics suddenly changes inside the ponds, at that time to use the nets and to separate the fishes from one place to another places. Sometimes that bunch jal, cast net, gill nets. Yes, are these types within period 2 to 3, 3 to 6 hours within. And other type, that is the identification of fertilization of eggs. Fertilized eggs appear transparent. What is the characteristics of fertilized eggs? Those eggs are successful for the breeding. Those eggs are successful for the spawning. Eggs appear transparent. That is the fertilized eggs. That is opaque, whitish. Yes, Indian major carbs above highest fecundity, and I have given that is the 18,000 fecundity of major carbs. That is the example Katla, and highest fecundity 2 lakh. And fecundity is depends upon the fecundity means egg laying capacity of the fishes. Egg laying capacity of fishes, and fecundity is depends upon the weight of the body of fishes. Body of the fishes. And what is the technique of induced breeding? Technique of induced breeding, all the fishes captured, breed in specific ponds. That is a tank also. Measure the species, that is the Katla, major Katla, Lebu, Mrigal, and that is the Indian major carbs. And some other species, that is exotic carbs, breed in the monsoon and July to September and October to November and so on. And this monsoon region mainly fully matured fishes and ripe breeder, and that breeders large ponds, and that is a transferred in the smaller ponds for the easy capturing the type of experiments to prepare as the breeding happa. Are these types, what is the figure diagrammatic representation of the happa? Isn't it? That is the I will show that is the structure of happa. Happa is nothing but that is specific type of the nets. Are these types of the happa? Yes. Happa, what is the materials required to the happa? You know, that is the bamboo sticks, 4 bamboo sticks, and that bamboo sticks are tied with the help of the ropes. Yes, ropes, 4 bamboo sticks tied with the help of ropes, and that is the and which may be that is the water below the net or happa. Happa is nothing but typical type of the net house, that is known as the happa. And these happa inserted or float inside the water, and some species are inserted in this happa. Some breeder, those ready for the breeding and spawning. Yes, which in this happa, that is a male and female also. Those are ready for the female and males for injected with the dose of described as earlier as in the dosage. Both the injection male and female introduced together in the breeding happa. Breeding happa is made up of fine meshes of mosquito net. The happa is prepared for the mosquito net cloth and fixed in the bamboo pools in the water, in flowing water. Yes, these types, that is the I have given breeding happa. Breeding happa as shown in figure. It is a rectangular box shaped net cloth are enclosed. All sides can be opened, only one above the roping cloth. Yes, while the injected the breeders into it, the breeders of happa variable in size. That is a size 1.83 into 0.9 to 0.9 meter for the breeding, and weight of the fishes below 1.63 kg, 1 kg below, or 1 or 1/2 kg. Those fishes are 1.5 kg to 4 kg at that time, above 4 kg, that is the size, second size 2.44 into 1.22 into 0.9 meter, that is breeding at the, yes. And third size for the breeders weight above the 436 kg, 4 4 kg above, that is the size of the happa, 3.662 into 1.83 into 0.9 meters. This types, that is the size of the happa. Happa is nothing but, that is the, as shown in figure, rectangular box container made up of the fine meshes boxes, yes, and net cloth. I have given information already. What is the hormones of the pituitary gland? With the advancement fish culture program, synthetic hormones have been preferred as the substitutes of the pituitary hormones. These are, which is the chronic gonadotropin hormones, name of the hormones of the substitute pituitary hormones, those prepared from the fishes and mammals. That is the chronic gonadotropin is the mammalian hormones, yes. Human chronic gonadotropism. I have given up till now the number of scientists. Those scientists studied about the hormones of the pituitary of the fishes and mammals. Both are the vertebrate with related, the fish and mammals of the hormone. Yes, prolactin hormones. These are famous hormones. It is very important hormones. Synaprin hormones. Yes. Pregnant mare serum, PMS. Pregnant mare serum hormones. Methyl testosterone hormones. Methyl progesterone hormones. Deoxycorticosterone hormones, DCA. It is also known as the DCA. Yes, these types, that is the hormones which is prepared for the vertebrates, the fish and mammals also. Yes, I have given information about the today we discuss about the induced breeding, historical background. Yes, historical background, some number of scientists studied in the foreign countries, our countries, and what is idea about the pituitary gland, and what is the aim of this pituitary gland, fertilized eggs, identification of fertilization eggs, and technique induced breeding, that is the breeding happa, and it's I have shown the figure, shown in figures, and which period is very important, that is the monsoon period is very important for the experimental, for the mature fishes netted in the large ponds for the breeding, male and females. Yes, and that types of the net which is a very important happa, as shown in figure. It is a rectangular in shape. I have given in detail rectangular shape. It is made up of the mosquito nets or cloths enclosed outside and prevent breeders and escape out. Yes, and open only from the above by rolling the cloth. While introducing the inject breeders into it, breeders happa are very valuable, and that is given the size of the nets. Yes, means those fishes at below 1 kg or up to 1 kg, that is the size first, means 1.83 to 0.9. Yes, and second size those fishes 6 163 kg to 4 kg, that is second size for 2.44 into 1.22 and 0.9 meter. The breeders weighing in between the 163 to 436. And third size view the 430 kg 4.36 kg. Above the third size of the net size of the happa 3.66 into 1.83 into 0.9 meter. At these types I have given the information about the induced breeding. Yes, now we discuss in detail remaining part, also discuss in next period. Yes. What maybe that is the difficulty about the induced breeding, you comments me, you give feedback, we discuss in detail. Thank you. Good evening.